My name's Phil Yules, and I'm a PhD student at the Baber Institute, and I'm one of the researchers here who works on epigenetics. So epigenetics is an extra layer of information built on top of the genetic code. Genes are made up of 4 bases, DNA is made up of 4 bases, and epigenetics is an extra layer of information which is heritable between cell divisions, which doesn't change those sequences, and it can affect the way that the DNA is read and used, and it can also affect the way it's physically compacted and organised within the cell. Epigenetics is a pretty new field. It's been first kind of proposed and started back in the 70s, but it's really started to pick up speed in the last kind of 10 years or so. The technology is starting to catch up with the ideas, and as more and more people get interested, as more expand out, it's becoming more applicable to lots of different fields. Disease, aging, cancer biology, lots of these things are now, we're realising, are affected by epigenetics. So the 2 key parts of epigenetics we're talking about here, first, DNA methylation, which is where you can take a normal cytosine base in DNA and you can add on a methyl group, a chemical group, and by modifying it like that, you can affect the way that different proteins might bind to the DNA. And so if you have a series of these bases, or an island of these bases, say in a promoter of a gene, if they're all methylated, then that can prevent the transcription factors or proteins from binding the DNA, and so stop that gene from being transcribed. And another way is we've got a vast amount of DNA within the nucleus of every cell, and it's packaged very, very carefully. And so by modifying the proteins which the DNA is wrapped around, that can affect how compressed and how accessible the DNA is, and so that in turn can affect whether genes are switched on or off. This is all useful because the key example of this is cell fate, where you start off with pluripotent cells which can turn into everything, but as they differentiate, their path in differentiation and their identity is fixed and remembered, if you like, by these epigenetic modifications of the DNA. One of the interesting sides of the science is pluripotency in stem cells, and something we'll talk about here are induced pluripotent stem cells, or iPS cells. And this is where we take a cell which is differentiated, which has had its identity sealed already, and then apply a number of factors to it to try and reverse these epigenetic modifications and see if we can give it pluripotent status again, make it into a stem cell. And this has got huge consequences, because if we can reliably do this, then there's lots of downstream applications. For instance, you could take skin cells from someone and reprogram them and make them grow into a new heart. You wouldn't need transplants or whatever. So there's a lot of excitement in that field at the moment.
Yeah, so the game we have on here is that people get a chance to play at being a DNA sequencer, and specifically they get to have a go at being a new kind of DNA sequencer that allows us potentially to look at epigenetic marks directly on DNA. So conventional sequencers that we have at the moment just sequence the letters in DNA. They can't look at any of the marks that are on top. But there's a new type of sequencer called a nanopore sequencer, which potentially can actually tell the difference between the normal natural DNA bases and those that have modifications on top. So the nanopore sequencers haven't actually been released publicly yet. There have been various papers that have come out showing that in theory you can do this, so that there's enough difference in the signal that these bases and modified bases produce as they pass through the nanopore, so that under the right conditions it might be possible to actually read this information directly. But the machines are due out basically any time, so they've been announced and they've demonstrated these, but no sort of scientists have actually got their hands on them yet to start doing this. So this is something that can really help us in the type of research that we're doing, if it does what it promises to be able to do.